Hello and welcome to our video on gametogenesis. In this video, I'll be telling us about what gametogenesis is, and then I'll talk about the first part of gametogenesis, which is spermatogenesis. And then in a second video, I'll look at gametogenesis in females, which would refer to as oogenesis. Now, gametogenesis involves the formation of gametes, haploid gametes, from diploid germinal cells through the process of mitosis. If you have not watched my video on cell division, you may want to watch that before this. Of course, from first principles, we know that during meiosis, which is better pronounced as meiosis, a diploid parent cell gives rise to haploid daughter cells. Same thing happens in gametogenesis. The parent cell, which is called a primordial germ cell, present in the gonad of the individual, undergoes meiosis to produce gametes that are haploid. During gametogenesis, there are many steps that take place, starting from the primordial germ cell until the gamete is formed. I'll focus first, like I said earlier, on spermatogenesis, and when I'm done with spermatogenesis, we'll have a second video where I'll look at oogenesis. Now for spermatogenesis, I'm going to talk about it in three parts. In the first part, I'll look at the stages or steps of spermatogenesis. In the second part, I'll discuss or describe the structure of the product of spermatogenesis. And then in the last part of this video, I'll talk about how hormones are involved in the process of spermatogenesis. So let's begin with the steps of spermatogenesis. Spermatogenesis occurs in a part of the male body called the testis. Now, this testis is known to be the male gonad. So females have their gonads and males also have theirs. The gonad is that organ, that point where gametes, sperm and eggs are produced. So the testis is the male gonad. What I've written there as testis is plural. The singular would have an I here instead of the E. So testis, that's singular. Now these testes are held outside the body of the male, inside what we refer to as the scrotum or scrotal sac. And of course, it is believed that this testis is held outside the body because the process going on within this testis, which is called spermatogenesis, is a process that is sensitive to temperature. It requires low temperatures. Extremes of temperature can distort that process. So it is believed that the testes are outside to provide the low temperatures required for adequate spermatogenesis. Now, within these testes, you have what we refer to as the seminiferous tubules. Seminiferous tubules. The seminiferous tubules are the main site within the testis where these sperm cells are produced. And of course, as this part of the name suggests, the tubule is truly like a tube. Now, on the wall of this tube, we have the primordial germ cell. So we call it the germinal epithelium of the seminiferous tubule. So this seminiferous tubule has germinal epithelium, that is the lining on its inner surface, and that is where you have the cells that are like parent cells to the gametes, the sperm cells that are like daughters in meiosis. So on the walls of the seminiferous tubules, there are plenty of what we refer to as the primordial germ cells, or in some contexts we just call them germ cells. Now, these germ cells are usually present in an individual from birth. So for a male that has the testes present, these germ cells are usually there under normal circumstances, but they do not begin to develop until that male reaches puberty. Why? Because their development is under the influence of certain hormones, and these hormones are not produced until puberty. So it is at puberty that what we're about to describe actually takes place. So in the presence of these hormones, the primordial germ cells will undergo a first stage which involves mitosis, another type of cell division. Now, mitosis is a type of cell division where you have diploid cells giving rise to fellow diploid cells. There's no reduction in number of chromosomes. So for the primordial germ cells, they are diploid, so I'll write 2N there. And when they undergo mitosis, it only gives them an avenue to multiply. So we say it's a multiplication division taking place there, so that from one cell you can get many cells. Those many cells you get after the multiple mitosis that will take place will be referred to as spermatogonia. And of course, as you already know about mitosis, diploid cells will give you diploid cells. So 2N will give 2N. That's mitosis. Now the spermatogonia will grow. So this is growth now. They would grow to become what we call primary spermatocytes. Primary spermatocytes, and these primary spermatocytes of course will still be diploid because it is just growth that took place there. Now for the primary spermatocytes, they have an alternative name. They are referred to as the sperm mother cell. The sperm mother cell. So if you hear of the sperm mother cell, that is a term that describes the primary spermatocyte. Now, what happens to the primary spermatocyte is the main spermatogenesis. So in a sense, spermatogenesis begins from this level because if you remember at the outset, I said that spermatogenesis involved the formation of haploid gametes from diploid parent cells by the process of meiosis. So even though there's mitosis at the start there, the main type of cell division giving rise to gametes is meiosis. So that for the primary spermatocytes or sperm mother cells, they would undergo the first meiotic division. Of course, like I said, you could go ahead and watch my video on meiosis and mitosis, because if you do, you'd realize that for meiosis, there are two stages. There's meiosis 1, the first meiotic division, and then there's meiosis 2. So meiosis 1 is what is taking place here, and when the primary spermatocytes undergo it, they produce what we refer to as secondary spermatocytes, and these ones are haploid. So we've gone from diploid to haploid. So while the primary spermatocyte is diploid, the secondary spermatocyte is haploid. Then comes meiosis 2. So during meiosis 2, so I put that here, meiosis 2. During meiosis 2, we have this secondary spermatocyte dividing to become what we call spermatids, which are also haploid. So the secondary spermatocyte becomes the spermatids by way of meiosis 2. So this is the first meiotic division, and then that's the second meiotic division. And at the end of both meiotic divisions, as you know from even secondary school, one cell would have given rise to 4 daughters. So from one primary spermatocyte, you expect a total of 4 spermatids. Now, these spermatids are like the mature sperm. They are like the mature sperm, but they are not the mature sperm. They need to undergo maturation. They need to undergo differentiation before they become the mature sperm cells called the spermatozoa. So the spermatozoa, or sperm for short, are the mature forms of the spermatids. And that process of the spermatids differentiating into spermatozoa is referred to as spermiation. So spermiation, oh sorry, that's not spermiation now, that's spermiogenesis, please. That's spermiogenesis. Spermiation is a different process. It has to do with the release of mature sperm cells from the Sertoli cell. There's what we call the Sertoli cell. It's a cell that nourishes the sperm cells. So the release of sperm cells from the Sertoli cells, mature sperm cells actually, is called spermiation. But spermiogenesis is right here. It is the last stage of spermatogenesis. So we can assume that from up there till we get to the spermatozoa, everything is spermatogenesis. In some other contexts, it is assumed that spermatogenesis is just from the primary spermatocyte all the way to the spermatozoa. But in any case, whether you look at it from there or from here, the very last stage of spermatogenesis, which involves the spermatids becoming spermatozoa, is what we refer to as spermiogenesis. So this is how the sperm cells are formed. And as of the time they become spermatozoa like this, they are usually attached to a cell, a giant cell that is on this wall. So you have the sperm cells like that attached to that giant cell. That giant cell has the role or the purpose of nourishing the sperm cells. It nourishes them. So from spermatid to spermatozoa, the maturation period, they spend it attached to this giant cell. This giant cell is called the Sertoli cell. So that's the Sertoli cell. And we mentioned before that the release of these mature sperms from the Sertoli cell is called spermiation. So those are the sperm cells, and this is the Sertoli cell. So that's about the process of spermatogenesis. I think it would be more appropriate to talk about hormonal control before we describe the sperm cell itself. Now, for hormonal control, there are various hormones that are involved in the process of spermatogenesis. These hormones include 1. GnRH. GnRH is called gonadotropin releasing hormone. Gonadotropin releasing hormone. And that hormone is produced in the hypothalamus. And then we have what we refer to as the gonadotropins. So the gonadotropins are FSH and LH. FSH means follicle stimulating hormone, whereas LH means luteinizing hormone. These two hormones are named based on their functions in the female. So in other words, I'm saying that males and females both have FSH and LH. Both hormones are present in males and present in females, but they get their names from their functions in females, from what they do for the female. Then, in a male, for luteinizing hormone LH, it is also called ICSH. ICSH means interstitial cell stimulating hormone. Interstitial cell stimulating hormone. So there are some cells that we call the interstitial cells present in the testes, and these cells are named after Leydig. So we call them the interstitial cells of Leydig, or we just call them Leydig cells. So Leydig cells are interstitial cells, and the work of LH will involve stimulating these interstitial cells, and that's why LH is called interstitial cell stimulating hormone. Away from that, the next hormone we are going to mention here is testosterone. Testosterone, that's the male hormone, although small amounts of it are also present in females. And then there's the last one called inhibin. So GnRH, like we said, comes from the hypothalamus. FSH and LH are produced in the pituitary gland, precisely the anterior pituitary gland, not the posterior. For testosterone, it is produced right there in the testis by the interstitial cells. So these interstitial cells, their function is to produce testosterone. So it means that when LH stimulates the interstitial cells, it is so that they produce testosterone. And then inhibin is produced by this guy, the Sertoli cell. Now this process from the primordial germ cell all the way down to the spermatid stage is under the influence of testosterone. So testosterone is the guy responsible for spermatogenesis. But this last stage of maturation of the spermatids into spermatozoa is under the influence of FSH. So FSH, or follicle stimulating hormone, will take charge of this last stage of spermatogenesis, which in fact we refer to as spermiogenesis. So we've said that FSH and testosterone are concerned with spermatogenesis. They are directly concerned. As for LH, I already told us its function, that it stimulates the interstitial cells to release testosterone. Then we are left with inhibin and GnRH. Now for GnRH, as the name implies, gonadotropin releasing hormone. It is a hormone produced by the hypothalamus and used by the hypothalamus to control the anterior pituitary, to stimulate the anterior pituitary to release the gonadotropins. So FSH and LH production is under the influence of GnRH. If GnRH is not produced by the hypothalamus, then you don't get FSH and LH. And if you don't get LH, it means you don't get testosterone. So I'd like to remove this diagram and show you something like a small pathway. That pathway will help you understand how these hormones work. So I'll draw something very simple here. Let's say we have at the top hypothalamus. This is the hypothalamus. Then this is the anterior pituitary. And then we have the testis. Beautiful. So something like this. Now the hypothalamus releases GnRH, and with this GnRH, hypothalamus is able to stimulate anterior pituitary to release FSH and LH. And then the testis will respond by releasing testosterone. And we said this testosterone is the one that will stimulate the production of the sperm cells. Now, if sperm production becomes too rapid, like there comes to be plenty of the sperms produced over a period of time, then sperm production can be slowed down either by testosterone or by a second route. Now, for testosterone, it is known to exert an effect on the hypothalamus. It is able to inhibit the hypothalamus such that by the time the testosterone stimulates, there is not stimulation now, inhibition. By the time it inhibits the hypothalamus, GnRH is not released from the hypothalamus. And as GnRH is not released from the hypothalamus, or its release is reduced, or the quantity released is reduced, then the anterior pituitary also will slow down on its release of FSH and LH. And with decreased stimulation of the testis, then testosterone levels will drop and sperm production will also slow down. That is called a negative feedback. Why is the feedback said to be negative? Hear what is negative about it. The initial problem was excess spermatozoa. And because there was an excess of the sperms, If the reply had led to the production of more sperms in the face of an excess, you would have called that feedback positive. But the feedback is called negative because due to an excess, there is a feedback leading to the production of less. That feedback is a negative feedback. So a negative feedback is one that produces a result that is the opposite of the trigger, but whereby the trigger is let's say an excess of something, and then a reply goes to a particular pathway and leads to further production of what is already in excess, then that is referred to as a positive feedback. In some instances, we talk about positive feedbacks and vicious cycles, but I'll leave all that for now. Now, that's about testosterone regulating sperm production, regulating an excess of itself of the sperms produced. But there is also inhibin, like I mentioned before. Inhibin is produced by the Sertoli cells. And remember that the sperms undergo maturation at the level of the Sertoli cells. So if the sperms are too much, the Sertoli cell is aware, so that the Sertoli cell can send out inhibin. Now, in the case of inhibin, it is able to attack, that's inhibit now, not necessarily attack. It inhibits the anterior pituitary. So it works at the level of the anterior pituitary, not the hypothalamus. And what does it do at the level of the anterior pituitary now? It causes it to reduce the amount of FSH and LH being released by the anterior pituitary, so that with the reduction in the levels of FSH and LH, the testis' own production of testosterone will drop. And when testosterone production drops, sperm production, spermatogenesis generally, will also reduce. So in that case, we say that the Sertoli cell releases inhibin to inhibit sperm production. But what if sperm production becomes too low? Then for this second pathway, all the Sertoli cell will need to do is send out less of inhibin. And when it sends out less of inhibin, then what we just described now is unlikely to happen, so that sperm production can increase. So everything is about this level. Whatever increases testosterone should increase sperm production and vice versa. So these are the hormones involved, and in summary we have said this guy brings about sperm production, then the other ones regulate it. For inhibin, it can slow down the production of testosterone, and then testosterone itself can slow down its own production. Those are the two pathways shown there. Then for GnRH, its work is to stimulate the anterior pituitary to produce FSH and LH. Then for FSH and LH, we said FSH stimulates spermatogenesis, while LH stimulates the production of testosterone from the testis. Now, to end this first video, we'll look at the structure of a sperm cell. What's a sperm cell like? A sperm cell is drawn like this. It's a very small cell, as a matter of fact, compared to the egg cell. The sperm cell is said to be about 2.5 micrometers long. So it's a very, very tiny cell, extremely small. And it's not surprising that it moves about 1 to 4 millimeters per minute. So in a 60-second period, it doesn't cover more than 1 to 4 millimeters per minute. And then also, the sperm cell is said to be produced at a rate of about 120 million daily. So every day, about 120 million sperm cells are produced. But these 120 that are released every day, it takes about 70 days to make them. So it takes about 70 days to make a particular sperm cell. And that means this production goes on every day, so that for every day you have about 120 million cells turned out, and whatever is turned out today must have had its production started some 70 days or so ago. So it takes about 70 days to get each sperm cell. A sperm cell on the average has a structure like this. I'll draw that very quickly, so we do not expect it to be very beautiful, but it will contain the necessary details. So a sperm cell is something like that. Okay, so we have this, we have the fibers at the center, and then we have a dense packing of mitochondria. Okay, so we're almost done with that structure. Yep, a sperm cell is something like this. This is called the head, so all of this is the head. Then this is referred to as the neck. And then there's a middle piece, so this is the middle piece. And then the rest of it is called the tail. In some texts, they talk about the tail piece and end piece, but we are okay with calling the rest of it the tail. Then coming to the head, within the head there appear to be two structures. This one is the nucleus, and of course it's not surprising because a sperm is actually a cell, so it has a nucleus. And then this is called the acrosome. Acrosome. So the acrosome is a sac present on the head of the sperm, and that acrosome contains some enzymes. We'll talk about the role of those enzymes when we come to the video on fertilization and organogenesis. So under fertilization, I'll tell you the role of the enzymes acrosine and hyaluronidase. Then for this middle piece of the sperm cell, okay, before the middle piece, what we're seeing here is the neck, and you see that there are two structures there. We call them the centrioles. So there are two centrioles at the neck. Then in this middle piece, like I said before, we have a dense packing of mitochondria. Why do we have so many mitochondria in the sperm cell? It's because the sperm cell does a lot of work, moving from where they are deposited during ejaculation up to the upper reaches of the oviduct. So to traverse, if you look at how short the distance that it moves in 1 minute is, you realize that it's a Herculean task for a sperm cell to go from the vagina all the way to the upper reaches of the oviduct where fertilization will take place. In fact, it's said that for a man, it's equivalent to swimming across the Atlantic. So because of the large energy requirements of that journey, the sperm cell has plenty of mitochondria to meet the energy demands. Then the tail, some call it the flagellum. It functions as a flagellum. It helps the sperm cell to beat its way up to meet the egg in the oviduct where fertilization will take place. Now, much of the sperm is empty. Even the head does not contain plenty cytoplasm, so that when this sperm meets an egg during fertilization, all it gives to the egg is genetic material. So for cytoplasm and food that the zygote eventually has, much of it will come from the egg. Now this tail we are looking at, this may not be very relevant. It is known to have some 11 fibers in a 9+2 arrangement. We talk about the 9+2 arrangement of fibers, something like 1, 2, 3, 4, 5, 6, 7, 8, 9, like that, and then 2 in the middle. So there's the 9+2 arrangement of fibers that is found in cilia and flagella of unicellular organisms, and that same arrangement of fibers is found here in the sperm cell. So we say that the flagellum of the sperm cell resembles the flagella in microorganisms in that it also has the 9+2 arrangement of fibers. So those are the parts of the sperm cell, and that's the structure of the sperm cell. We've talked about its length, 2.5 micrometers, and so on. I'll spare you all the other details, unnecessary details this may bring our way. But for now, I'll pause. I'll let this video end here, so that in the next video, I'll look at the parallel process called oogenesis. So I'll see us after the break in the next video.